Friends, welcome to the third session of immunity and autoimmunity, immune dysfunction for the physician. I'm very grateful to Dr. Debashish, our previous head of rheumatology and the previous head of medicine too, my chief, for agreeing to arrange this beautiful session. We have benefited enormously from the first two sessions, and today we are going to hear again Professor Ramnath Mishra and Dr. Sita Naik, and I'm eagerly looking forward to it. So over to Dr. Debashish sir. His brother is not well, but still sir has uh, kept his word for us and has arranged this. So we are very grateful and pray your brother gets well soon. So over to you, sir.
Thank you. Uh, Welcome everybody. Without wasting much time, since the audience is already familiar with the speakers, Professor Mishra is the ex-dean and ex-head of clinical immunology and rheumatology in SGPGI Lucknow. And right now he's the research dean of KIMS Bhubaneswar and also the head of clinical immunology and rheumatology. Dr. Mishra has enormous experience and knowledge in the field of immunology, has been my teacher and my mentor. Dr. Mishra was also spent a considerable period in the UK with stalwarts in immunology like Dr. Ravinder Maini and others in Kennedy Institute of Rheumatology. Sir, welcome. In this session, Dr. Mishra sir will be covering on antigen presentation and autoimmunity. And I think sir will also cover the leftovers from the last talk, that is on phagocytosis and autophagy and so on. And after sir's talk will be Professor Sita Naik's talk. She was also the Dean of Head of Department and one of the finest immunopathologists in the country, my teacher, and she was also the MCI governing body, and she's involved in multiple academic immunology and social activities. So Madam is also a very good, what shall I say, I wouldn't call activist, but Madam keeps writing on the blogs, on her blogs, on various contemporary issues. Welcome, Dr. Mishra sir. Sir, please, over to you.
Thank you, Debashish, and our other host. It is indeed a pleasure to come back again, and my talk would be on antigen presentation and autoimmunity. Well, it's a very important talk in the sense that a lot of diseases that we deal with, the mechanistic explanations would be clarified in my presentation, at least most of it. In order to understand the immune system and how tightly regulated and specific it is, here are the molecules that are involved in it. These are key molecules for immune response. Many of you would know them, that they are important molecules for rejection or transplant rejection or mismatching. These are MSC molecule 1 and 2. On the left side is called the class 1 MSC molecule, which is actually a single chain having 3 domain alpha 1, alpha 2, and alpha T. Between domain alpha 1 and alpha 2 is the antigen binding cleft or groove. It is associated with a small molecule that is beta 2 microglobulin. As you know, everyone's HLA molecule is specific to the same individual. We have 3 A, B, C. We have several antigens of A, several allotypes of B and C, and you may be familiar that certain HLA molecule are associated strongly with some disease. The class 2 molecules have got 2 polypeptide chain, the beta chain and the alpha chain, with each having two domains, the alpha 1, alpha 2, and beta 1, beta 2. And the antigen binding domain is between alpha 1 and beta 1 domain. And these chains are connected with interchain disulfide bond. In this context of the HLA molecule, I will be talking about antigen presentation. How is antigen presented and recognized by the immune cells? It is the T cells that to which the antigens are presented. And in the cell-mediated immune response, macrophages present the antigen of phagocytosed microbes to effector T cells. And there, the MSC class 2 molecule is regulated. And these are presented to the CD4 T cells, CD8, CD4 T cells. The humoral immune response, which is carried out by the B lymphocyte, again, it can present as an antigen presenting cells to present to T helper cells, which gives the help to the B cells to produce antibody, class switching, and more finer antibody. In our body, all nucleated cells have class 1 molecules on them, but class 2 molecules are only present on the antigen presenting cells. And which are the antigen presenting cells? The macrophage, the dendritic cells, and the follicular dendritic cells. They are specialized cells, and they only have class 2 molecules expressed on them, and that too when there is a situation of alarm, or that is infection or a foreign microbe that they see. There are two pathways of antigen presentation, the class 1 and the class 2. Class 1 antigen presentations are all antigens which are present inside the cell in the acidic vesicular compartment. Class 2 are antigen present in the cytosol, which are phagocytized by microbes. So those microbes, when they are phagocytized and processed, these antigens are presented by the class 2 molecule. So let's see examples of both. Presentation by class 1 MSC. These are the source of cytosolic antigens of viral encoded protein, such as these days it's COVID. Any virus that are present, virus usually have cellular location, and these viral encoded proteins, tumor and intracellular bacterial, these are proteolytically degraded by a protein complex called proteasome, and the peptide that are generated is carried from the cytosol to the endoplasmic reticulum, and where the assembly of peptide with class 1 molecule occurs, and then it is surface expressed. This is a diagram, a cartoon of it. So virus in cytoplasm, they synthesized viral protein. These proteins are ubiquitinated, and the proteins, these proteins are taken up by the proteasomes to get degraded. It's a very powerful enzymatic enzymes are there in the proteasome that digest the protein into peptides, and these peptides, with the help of a molecule called transport associated protein, gets uh, into the endoplasmic reticulum. And in the endoplasmic reticulum, the class 1 MSC molecules are synthesized with the beta 2 microglobulin. And these molecules called the ERAP, it trimmed the peptide so that it can fit in the groove between the alpha 1 and anti 2. And finally, it comes to the Golgi apparatus through the exocytic vesicle and presents the molecule to CD8 to carry out the cytotoxic T lymphocyte. Ultimately, the cell, which is presenting the antigen, either the viral or the tumor antigen to the CD8 T cells, it results in destruction of the cell. There is cytolytic death of the cell because the cell presents a foreign bacteria or a tumor protein. The pathway is somewhat different for class 2 presentation. Here, the proteins or the microbes is taken into the vesicular compartment. It is processed through endosomes and lysosomal enzymes. And the class 2 molecules are again synthesized in the endoplasmic reticulum, and where there is association of class 2 molecules with peptide, and the MSC2 peptide complex surface on the cell. So here the protein or the microbe is taken, and then lysosome has several proteases. These are engulfed in endosomes. There is fusion of endosome and lysosome, and in the simultaneously in the endoplasmic reticulum, where the class 2 molecules are synthesized to chain the alpha chain and beta chain, it has a protein which is an invariant chain, which is called the clip protein. This protein enters the Golgi apparatus into the exocytic vehicle, and then this peptide, the clip protein is hydrolyzed by a molecule called DM, and then the peptide get assembled into the MSC class 2. Unless the peptide or antigen is presented in the context of an HLA, no immune response can occur. And here it presents the antigen to the CD4 T cells, which is the helper T cell, that gives signal to the B cell to produce antibodies. What about the proteasomal degradation? They are large multi-protein enzyme complex stacked in a cylinder fashion, and it performs the basic housekeeping tasks of removing damaged and misfolded protein by ubiquitation, which is recognized by proteasome. This table gives a comparison between the two pathways of MSC presentation. As you see, in class 2, the peptide is docked between two alpha chain, the alpha 2 and beta 2 chain, whereas in the class 1, it is between the alpha 1 and alpha 2 domain of one chain. There isn't a second chain. Now, what are the cells that have class 2 presentation? It is the dendritic cells, the mononuclear phagocyte, the B lymphocyte, endothelial cell, and thymic epithelium. But all nucleated cells have class 1 molecules. Class 2 molecules can be overexpressed in a situation of need when there is a danger to the body. The responsive T cell is the CD4 T cell, and here it's the CD8 T cell. And the source of protein for class 2 is endosomal or lysosomal protein, mostly internalized from extracellular environment, whereas for the class 1, it is the cytosolic proteins that are mostly synthesized in the cell and may enter cytosol from phagosome. And enzymes responsible for peptide degradation are the endosomal and lysosomal proteases, such as the cathepsin. And here the unit is called proteasome. The site of peptide loading is a specialized vesicular compartment. Here the peptide loading occurs in the endoplasmic reticulum. The molecules involved are the chaperones in ER, the invariant chain or the clip molecule, the Golgi, and the DM molecule. Here it is the chaperones and the tap in endoplasmic reticulum. With this basic information, I move on to how autoimmunity is checked in the body. Means how do we get tolerance to cell proteins? It starts in the thymus itself. That is called the central tolerance, and therefore we don't respond to our own protein. The immature thymocyte, which has CD8 and CD4 markers, and the T cell receptor, it first undergoes a positive selection. That means only those which has self-MSC molecule, they are only selected. Other HLM molecules, these cells, they undergo death by apoptosis. And after the positive selection, then comes a negative selection. That means cells which have the TCR, which are recognizing self-antigens, are deleted. So at the thymus, when cells come out, they have self-HLA and an anti-TCR or a antigen receptor that is not recognizing to self-antigens. So this education in the thymus is otherwise called as the mechanism for central tolerance. Now, just to give you how an antigen presenting cell presents to the T cell, we heard that the antigen or the peptide, let us say, is to be presented in the context of a HLA molecule to the TCR that is present on the T cells. And you can realize that there are about 10 million different type of T cells requiring, all have their own specific antigen receptor on them. To have an efficient activation of T cell, we need co-stimulatory molecule. And this is engagement of two molecules, the CD28 present on the T cell and the B7 present on the antigen presenting cells. When the antigen is recognized by the T cell, we need the co-stimulation. If there is no co-stimulation, the T cell is quiescent. But in presence of a positive co-stimulation, the T cell gets activated, producing IL-2. Of course, the antigen presenting cells in contact with pathogens or damp or PAMP, they get activated, and that is when the class 2 molecules are expressed. even if suppose there is some leaky T cells coming out of the thymus who have the potential to react with self antigen, but there are peripheral tolerance mechanisms or mechanisms to inhibit these self-reactive T cells. One of the mechanisms is called immunologic ignorance. That means these self-antigens, they are never expressed to the antigen-presenting cells. Something like the example that is commonly given is enzyme of the proteins of the retinal protein antigens. There is an effective barrier so that the peripheral lymphocyte, peripheral antigen presenting cell doesn't come in contact. There are antigens in the substance of the central nervous system, and you know there is a blood-brain barrier. Even the testicular antigens, they have anatomical barrier. But if there is an injury and this barrier is compromised, these antigens can come into the peripheral circulation and mount an immune response. This is then if there is autoreactive T cells in the periphery, they can be destroyed or damaged by body's peripheral system check. They expresses fast ligand, and these cells get deleted. The autoreactive T cells can have inhibitory co-stimulations resulting no T cell activation. And then we have regulatory T cells which actively suppress an activated T cells, and this is mediated by IL-10 and TGF-beta. So there are a number of mechanisms that are that make the individual foolproof against host antigen or self antigen, and this ranges from not seeing the antigen. If they are present, then the cells are deleted, even if they escape the central mechanisms. They can be directly inhibited through co-stimulatory molecule or indirectly through the regulatory T cells. So what are the ways self-tolerance can be broken? You can have sequestered antigens released, such as the eye antigens in case of an trauma or infection. If they are released, you can have immune response to them. You can have neoantigen, which the body has never seen before, such as the drug and the hapten. Sometimes there is polyclonal stimulation. Clone of lymphocytes, such as the B cell, they get stimulated and produce self-reactive autoantibodies. There can be aberrant expression of class 2. I told that class 2 expression is tightly regulated, but if they overexpress themselves in the APC, then one can get autoimmune disease. We'll give an example later on. There is a danger hypothesis, mean there are, if a microbes or a tumor antigen is seen by the body, by the macrophage, it starts expressing class 2 molecule and producing interferon gamma, so that a set of cells which are nearby, they get activated. And chronic activation of this can produce autoantibodies, and one can have molecular mimicry. So a resting tissue APC is a quiet cell. Even if produce self-antigen, the T cell doesn't respond to it. But if there is a microbe and the MSC has produced a self-antigen, the APC gets activated and the co-stimulatory molecule, as I mentioned, B7 and CD28 are expressed, and you possibly can get a self-reactive T cell. Here, it is the microbe that has activated the APC to produce co-stimulation. Sometimes what has happened, the microbe produces the APC produce microbial antigen, and there are T cell which is directed against the microbe. But because of molecular mimicry, this T cell, they start reacting with structures of the body which have got similar sequences like the microbe. There are examples of several protein molecule of infectious organisms which have got molecular mimicry or sequence similarity with host molecule. The first one says cytomegalovirus with HLA-DR molecule, poliovirus with acetylcholine receptor, and you would have realized that either the virus or the bacteria, they are all made of those amino acids that our proteins are made of. So it is not surprising that papillomavirus can have similarity with insulin receptor. And the theory here that if you get infection with a papillomavirus, we can possibly get damage to the pancreas or the beta cells. And similarly, Klebsiella pneumoniae organisms have got molecular sequence similarity with HLA-B27 molecule. And I mean, this is a small list. The list can be huge. This is a very attractive theory that you get measles and you can have multiple sclerosis. But in practice, or pragmatically, this has not been proved. Means, except for our story of rheumatic fever following streptococcal infection, we get cardiac lesions. There are not many other diseases which have got convincing evidence that it is the molecular mimicry that cause self-reactivity. Here I would like to bring in one concept of autoimmunity and autoimmune disease. Autoimmunity is the presence of T cells which are reacting to self-antigen, or B cells which producing autoantibodies that is reacting to self-antigen. Autoimmune disease is where there is inflammation and damage to the self-organ. Autoimmunity is widely prevalent, but autoimmune disease are less. And it is believed that there are several steps required from autoimmunity to autoimmune disease. So even if you have some autoantibodies or self-reactive T cell, it doesn't add up that you would end up having an autoimmune disease. Autoimmune diseases are traditionally viewed as organ-specific autoantibodies or non-organ specific autoantibodies. And these are examples of disease here, like the Addison disease, you get damaged adrenal cells, autoimmune hemolytic anemia to the RBC membrane protein damaged by autoantibodies. Wood-Passer syndrome, it's the renal and lung basement membrane. Graves' disease is thyroid stimulating hormone receptor. Organ specific means it is in Graves' disease, thyroid gland is affected. It doesn't affect any other organs or tissue system. ITP, you get platelet destruction because of autoantibodies. IgGM, you get pancreatic beta cells, and therefore these are categorized as organ-specific autoantibodies. And on the right hand side, of course, Addison disease is caused by autoantibody, whereas Hashimoto's thyroiditis is caused by T cells, like the IgGM. And you can have pernicious anemia where the intrinsic factor, autoantibodies to intrinsic factor is there. These are examples of organ-specific autoantibodies. Whereas non-organ specific autoimmune disease affect many organs. We have examples of ankylosing spondylitis, multiple sclerosis, rheumatoid arthritis, scleroderma, Sjogren's, and SLE. It can affect the kidneys, the lungs, the joints, the skin in SLE. So it is not specific to one organ. The way to understand how autoimmunity or how this causes disease, it's usually the mechanisms are four. It's called hypersensitivity. That means inappropriate response of the body to self or foreign antigen. We have four types: type 1, type 2, type 3, and type 4. The type 1 hypersensitivity, the example is that an anaphylactic reaction. If you have preformed antibodies on exposure to allergen, the eosinophil and mast cells releases very powerful anaphylatoxin substances which can cause death. And we all know of a classic example is that of someone who is hypersensitivity to penicillin group of drugs. A single shot can kill the patient. Type 2 hypersensitivity are all those examples where the antibody binds to target antigens. In type 3, it is the antigen and the autoantibody complexes that get deposited in the tissues and cause damage. Type 4 is a delayed type of T cells that gets localized in the organs or the tissue that causes damage. This is an example of autoimmune hemolytic anemia. You can see that these RBCs, they had been coated with antibody to RBC that's seen in autoimmune hemolytic anemia. And when they go through the spleen takes off that antibody, and these cells are the red cells, which is normally like this. It takes a different form. These are called as spherocytes. This is an example of a type 2 hypersensitivity. So the antibody, which is there on the self antigen, is taken up by the phagocyte through the Fc receptor. And the complement is a good opsonized cell, which makes the cell palatable for phagocytosis. And the end result is that the self cells, such as the RBC or the platelet, they get destroyed by the phagocytes. And here is an example of the Goodpasture syndrome, where the antibody lines the autoantibody lines the glomerular basement membrane, and you get a similar effect in the lungs. Here, in the immune complex disease, this is the antibody that gets deposited, and that it causes complement-mediated damage to the local site. So antibody can, these are antibody-mediated, where the antibody deposits in the cell, like the glomerular basement. The Graves' disease is a very unique example. Here, there is autoantibody to the TSH receptor. And this autoantibody, which is acting on the TSH receptor, it's a stimulating antibody. That means it stimulates the hormone synthesis. It does the work of TSH itself. And therefore, you have excess thyroid hormone, and all the manifestations of Graves' disease are explained by the fact of inappropriately high thyroid hormone. And this is mediated by an antibody to TSH receptor. Whereas this is an example of blocking autoantibodies as seen in myasthenia gravis. At the neuromuscular junction, you know, acetylcholine is responsible for muscle activation. And in myasthenia, you have autoantibodies to these acetylcholine receptor. Therefore, when these receptor binds to, these antibodies bind to the receptor, the acetylcholine have no place to act, and you get neuromuscular paralysis or muscle paralysis. That is the explanation for the symptoms in myasthenia. Here, the example is that of a blocking autoantibodies. And this is a patient with SLE, which has a young lady with Miller rash. And here, this is an example of a non-organ autoantibody, non-organ specific disease, autoimmune disease. Here, we have plenty of immune complexes, which are antibodies, which are targeted to the antigen, and they are circulating. And this, they get deposited in the small blood vessels of the skin and the kidneys, perhaps the retina, and then it induces inflammation, attracting neutrophils, causing damage to the capillary and surrounding tissues. So therefore, in immune complex disease, as the immune complex rises, the antigen titer decreases, and the complement is deposited, and you have at the immune complex get deposited in the joint, kidney, and vascular, and you get the manifestation of arthritis and nephritis. And as after some days, there is only free antibody, there is no symptom. And this is how the immune complex is deposited in the kidney in an immune complex mediated disease, and you get a type of lumpy and clumpy immunofluorescent deposit. You can see the deposit even in electron microscopy. In delayed type of hypersensitivity, that is mediated by T cells. The T cells which sees the tissue antigen, when it, the cytotoxic T cell, it recognizes self T cell, it causes tissue injury. And we have example of, this is a Crohn's disease, the diabetes mellitus, the subacute thyroiditis, all these are mediated, and even rheumatoid arthritis are mediated by delayed type of T cells, which are reacting against self antigens. The example of this is also multiple sclerosis. What are the experimental animal models of autoimmune disease? We have spontaneous autoimmune disease, such as the NOD mouse, which has got IgDM. The inducing antigen is not known. The F1 generation of a cross between NZB and NZW mice is a classic example of SLE in a mice. And in obese trained chicken, you get Hashimoto's thyroiditis. Here, the inducing antigen is thyroglobulin. You can experimentally induce autoimmune disease, such as the experimental autoimmune myasthenia gravis through acetylcholine receptor. You can have an experimental autoimmune encephalitis by injecting myelin basic protein with a proteolipid protein, and you can have rheumatoid arthritis in a mice or picture similar to rheumatoid by injecting M. tuberculosis. So this gives us an example or how we can study the autoimmune disease in the mice and also develop drugs to work in this. The proof of autoimmune disease, if it is T-cell mediated, has to be very robust to call it that the disease phenomena is due to T cells or antibodies. This is way, as you will know, a Cox hypothesis where you prove microbe is the basis of the disease. Similarly, for example, MVP stands for myelin basic protein. If it is given with fluid adjuvant, you get T cells which respond to MVP, and ex vivo, you can isolate the clone of T cell and then inject to a normal rat. And if you get the disease, then it is a direct proof that it is because of autoreactive T cells. I told you in the central tolerance that all the self antigens, T cell reacting to self antigen, are normally deleted. There is a molecule called AIRE, which is responsible of expressing peripheral tissues in the thymus to self-reacting to the thymocyte. If this molecule is defective, then all the peripheral antigens are not seen by the thymocyte, and you get a large number of cells which are self-reactive B cells or T cells. Similarly, if there is defective regulatory cell because of a molecule called FOXP3, which is an integral molecule of regulatory T cell, you do not have the ability to apoptosis self-reactive T cells. And these self-reactive T cells, they escape the thymus and cause a syndrome which is called as immune dysregulation, polyendocrinopathy, and enteropathy. This is called IPEX because there is defect in the regulatory T cells. These are examples of genetic defect that causes autoimmune disease. And even if you have fast ligand, are effective mediator of apoptosis. And a large number of self-reactive T cells undergo apoptotic death because they recognize self-reactive T cells. However, if you have fast deficiency, these cells get escaped and cause autoreactivity. So in summary, we have many explanations for autoimmune disease. You can either have release of sequestered antigens, these antigens normally which are sequestered or hidden to the immune system if they get exposed. If you have inappropriate MSC expression on non-APCs, they start presenting self-antigen, and you can have APC with cross-reacting antigen, molecular mimicry, and there are several ways tissue damage can occur, either directly by the antibody, as shown here, through polyclonal activation, or you can have a T cell, which is cytotoxic to self antigen. These are few slides of my talk that was not covered in the last slide, and Devashish told me to uh, have this concept of cell death in you. There are two types of cell death. One is called programmed cell death or apoptosis, and a non-programmed cell death called necrosis. Apoptosis is a very controlled phenomena. It is always so many cells in the body, millions of cells undergo a programmed cell death. And it can be also due to trauma, toxic stress, self-renewal, aging, and development. Necrosis is uncontrolled. When there is a situation like trauma, toxic stress, and infection, the cell undergo necrosis. The morphology is radically different. Here, in the cell membrane, there occurs blebbing and condensation and fragmentation of the nucleus. Whereas here, inside the cell, there is organelle and cell swelling, loss of membrane integrity, and release of extracellular contents. In apoptotic cell death, the membrane, cell membrane doesn't get affected. The signaling pathway are through several enzymes. There are two set of pathways, the intrinsic and the extrinsic pathway. Both these pathways converge on an enzyme complex called caspase 3, 6, and 7, 8, and 9, and this mediates the cell death. Here it is unspecific, and there are a lot of cellular enzymes, proteolytic enzymes within the lysosomes, and they are packed in vesicles. They cause disease. This is in apoptosis, there is release of cytochrome C, interaction with the BCL2 protein, it's an anti-apoptotic protein, and mitochondrial dysfunction. This requires energy like ATP. Here there is no energy. There is mitochondrial dysfunction, collapse of the mitochondria. Apoptosis doesn't end up in a pro-inflammatory response. Here it causes a pro-inflammatory response. The damped molecules are released by this, and in necrosis, this is two pathways of apoptosis. One is the intrinsic pathway, and one is the extrinsic pathway and the intrinsic pathway. The extrinsic pathway are mediated by Fas ligand and Fas, and the TNF ligand to the TNF. This causes a death domain protein activation, and which is normally inhibited by this, and it results in caspase 8 activation, and then caspase 3, and the cell undergoes apoptosis. In here, in the intrinsic pathway, there are BH3 only proteins are there, which inhibit the BCL2 and directly cause the mitochondria to release cytochrome C, and that activates APF1, which cleaves pro-caspase 9 to caspase 9, and that leads to activation of caspase 3. You would remember that in complement pathway, it is the C3 that is central to both alternative and classical pathway. Here it is caspase 3, both two. Phagocytosis is, you are well aware of it, all the microbes of tumors, the antigen presenting cell or the macrophage, it takes up the cell and into the phagosome, where it is digested into smaller peptide. That is how body's first line of defense mechanism. There are selected receptor for phagocytosis, and opsonic phagocytosis is done by the Fc receptor family of the immunoglobulin, the complement receptor, and alpha 5 beta 1 integrin. And the ligands for these are the antibody with the antigen, the complement opsonic and fibronectin. Non-opsonic phagocytosis are dectin-1, macrophage receptor Marcos, scavenger receptor A, and integrin. And these are the various ligands for it. And non-specific phagocytosis can also be activated by the TLR ligands binding to the TLR molecule. Autophagy is defined as a process that delivers cytoplasmic material of endogenous or exogenous to the lysosome for degradation. The basic purpose of autophagy is to enable the cell to adapt to environmental stress, and it very much conserves the energy. It renewed or recycled protein. There are three main forms of autophagy: macrophagy, microphagy, and chaperone-mediated autophagy. I will stop here. Um, the death pathway, and would take on questions.
Thank you, sir. Thank you for
Ma'am,
explaining it so well. Uh, so should we take the questions at the end, Dr. Tambu, or should we take now? Sir, sir, you are here till the end of the session?
No, I have to go for a meeting at 4 o'clock.
Okay,
But
fine.
and today it is in a different building.
Okay, sir.
So I'm doing
Okay,
it
sir.
from home.
Sure, sir.
Maybe I would ask Mohan to send me the questions. I can
Okay, sir.
write it down and
Yes, sir. Okay,
email
sir.
him to him.
Okay, sir. That
sure,
would be
sir.
fine. Sure, sir.
Thank you, Devashish.
Thank you, sir,
And
for coming.
good afternoon, madam. And hopefully your lecture will fill in all the vacancies or all the lacunae in my lecture.
Thank you.
So before
Thank
we
you,
start,
sir.
thank you very much, sir. We are very grateful that you came and took
Yes, sir.
just before another class. Thank you.
Thank you,
Uh,
sir.
Before we move on, I just like, sir, with your permission, I want to introduce Dr. Anbalagan Pongavanam. He is the MCI, Tamil Nadu MCI inspector for our session. And because we are getting MCI credit points for people who attend the session. So welcome to Dr. Anbalagan. Thank you for joining us, sir. So we welcome you. His request is that if people could register when you log in for these sessions, if you could log in with your original name as registered under TNMC, it will be easy for future giving of certificates. So with that brief welcome to Dr. Anbalagan, I'll hand it over back to Dr. Danda to introduce Dr. Sita Naik.
Thank you. We are delighted that the MCI inspector is there watching our session. Now, the next speaker is Professor Sita Naik, who is an eloquent speaker. You have heard her before. Madam is an excellent communicator. And we were DM student, Madam had conducted course on scientific communication. And when I was in Australia, I had a booster dose of that, and I found it much easier as Madam has taught how to communicate both verbally and in written for all the DM students in SGPGI when I was there. Some of you may not know, Madam actually belongs to this part of the country. Madam originally hails from Palghat in Kerala. So I think, and Madam is actually a Tamilian from there. So nice, Madam. Welcome, ma'am. Please go ahead.
On SLE, what is the current understanding, sir? Is it antigen, antibody, T cell, and or what is the current understanding of the pathophysiology, sir?
Right. So SLE basically is a prototype immune complex mediated disease, and that is that holds good forever. But what has come, as Sir has mentioned, how circulating immune complexes can be highly inflammatogenic. But what has come recent years is the presence of the interferonopathies, The interferonopathies or overactivation of the interferon pathways is one important mechanism in lupus autoimmunity. The other is a defective autophagy. As Sir has shown, the altered autophagy is another mechanism in lupus. So these are the three things: circulating immune complexes, the interferonopathies, and autophagy mechanisms are the ones which are primary immune mechanisms in lupus. Lupus also has a Th17 arm, little bit active like other autoimmune diseases, but predominantly these are the three important mechanisms for lupus autoimmunity affecting multiple systems.
In the meantime, sir, can I ask some questions, sir?
Yes, sure, sure.
You had mentioned SLE, there was three mechanisms of
Yes,
autoimmunity. For
yes.
each of those mechanisms, is there specific now and treatments? And if there are, which situation should
Yeah.
we use them? Is it focused like that or?
Yeah. So I think for our real life clinical scenario, still the circulating immune complexes pathway holds very good. SLE is a museum of autoantibodies, and the autoantibodies are non-specific. They are not, as Dr. Mishra said, they are non-organ based, and that is why we get double stranded Smith and RNPs and a whole lot of other antibodies in lupus.
By the time the mammals and humans arrived, there was this huge external um, world, universe of organisms which was around. And to survive, we had to learn to deal with them. And therefore, some of them we learn to live with within ourselves. We have a whole flora inside us, and now we know so much about it, our gut flora. But some of us, some of them we could not live with. They were not so, we were not so welcome to them, and they were not so kind to us. So the immune system kept developing itself to get rid of these invading agents. So if you see the, um, the evolution of the immune system, and people have studied this going way back to the very, very earliest organisms. The first cells or immune cells that have been discovered are simple macrophages. In fact, a unicellular organism like the amoeba ingests external agents, but without any receptors. But when you come to early parts of the amphibian world, like lampreys, they have very simple macrophages with very poorly developed recognition molecules, but they do have those by which they can ingest viruses and bacteria, and they have enzyme systems which can break them down. But as organisms became more and more complex, the infections became more and more difficult, we developed more and more arms or various mechanisms by which we could fight these infections. So the present immune system in the mammal is what we are talking about. And although it has evolved to deal with immune external agents, we are not always competent, or our immune system is not efficient in dealing with them. And that is, so as we saw in the past, to just very, very quickly recap the basic outline of how the immune system works. It's basically designed to deal with external agents. There is an early defense, which is what we call the innate immune response, which consists of macrophages and cells of the mononuclear phagocytic system, the dendritic cells, which have a large a variety of non-polymorphic cell receptors, which between them can recognize a whole range of external infective agents as non-self, and therefore respond to them, right? And when this happens, and these are the large, we saw it last time, a whole array of these very, very smart molecules on the cells of the mononuclear system, which help in recognizing self and non-self. So Dr. Misra just finished a long lecture on how the failure of this self-non-self recognition is what causes autoimmunity. But the competence of this self-non-self recognition mechanism is what helps us to mount an immune response against foreign agents, which is predominantly the infective agents. So once the innate immune response is activated and it is efficient and gets rid of an infection, everything is fine. But in the failure to deal with an infective agents, especially when infective agents are competent and are able to multiply, as in the case of viruses, for instance, then the innate immune response has the capability to bring in reinforcements in the form of the acquired immune response or the adaptive immune response. So the cells of the mononuclear phagocytic system then present these antigenic peptides from these infective agents, which activate the T cells and the B cells, and then we have this whole range of actions which end in both activation of the B lymphocytes, which help in antibody production, the CD4 T lymphocytes, which produce cytokines, which cause the inflammation and help in the stimulation of the B cells, and the cytotoxic CD8 T cells, which kill infected cells which are hosting intracellular pathogens. So you have a whole armamentarium of mechanisms by which you can deal with different types of infectious agents. And in addition to this, in order to maintain ambient or a quiescent level of activity within the body, there is also a system of regulation, so that once an immune system is activated by an infection, it doesn't just keep on going in an unlimited fashion, but is controlled, and therefore, as and when the requirement of the external agent is dealt with, then it can be down-regulated and brought back to basal level. And this is a very important activity, because it is during this down-regulation and the control of infective agents, the contraction of the expanded immune cell repertoire. During this phase, there is a generation of the T memory cells and the B memory cells. These are very essential, and this is what allows the immune system to deal with a subsequent exposure to the same infection in a more vigorous and accelerated fashion. And this is what we call immune memory. So obviously, infective organisms, as we are all aware, belong to two broad categories. There are the extracellular organisms and there are the intracellular organisms. The extracellular organisms, by and large, are the bacteria, and these stimulate because they are outside, they are in the circulation or in areas wherever the infection takes place. They stimulate both the B cells as well as the T cells, but predominantly and through the activation of antibodies, also the complement system. So these extracellular organisms are neutralized, or they are coated with antibodies and then ingested by phagocytes so that they are eliminated, right? Now, they also are a very important role for complement in this process, because when these organisms get coated with complement, as we see here, the phagocytic cells have very potent complement receptors, and those bacteria or agents which are coated with the antibody and complement then are ingested through these receptors for complement. So this is a very efficient way of mopping up the extracellular bacteria, and macrophages are very, very powerful enzyme. They have very powerful enzyme mechanisms by which they can lyse and destroy these agents. Now, some of these bacteria are also directly lysed. In this whole process, the CD4, or the T cell arm of the immune response, has a role, because the cytokines that they release, obviously, as we've already seen, they are needed to provide the help for plasma cells, for B cells to become plasma cells and produce antibody. And also some of the cytokines help in activating the macrophages. There are macrophage activating cytokines, which then help in further ingestion and lysis of extracellular agents. So extracellular organisms, maybe a staphylococcal infection, or it may be pneumococcal infections, they are dealt with in extracellular spaces. And the reason why we get reinfection often with the same organisms, for instance, staphylococcus. In your lifetime, you may have multiple events of staphylococcal infection, because since the immune response is not, especially the T cell, is not activated to its full potential, there is no expansion of a large number of B cells and T cells, and there is no generation of memory. So the host does not recognize this next episode of staphylococcal infection and will remount a similar response each time it sees the same set of bacteria. But this response is efficient enough to get rid of these infections most of the time. And in the past, or till now, we have been also fortunate that the large repertoire of antibiotics that we have deal with these organisms, and therefore that we are able to look after most of these extracellular infections. But it is the immunity to the intracellular pathogens which continue to remain a challenge. So what are intracellular pathogens? We have some bacteria like the mycobacteria, salmonella, etc. We have DNA viruses, hepatitis B, papilloma, CMV, EBV, etc. We have RNA viruses, the most notorious of them being HIV, hepatitis C, and now, of course, the coronaviruses. So these are pathogens which all have ingestion mechanisms. They use certain receptors on cell surfaces to slip into the cell. Now, once they slip into the cell, they also may, in many cases, have the capability of neutralizing various antibacterial or antiviral agents that may be within the cell. And this is why some of these viruses and bacteria are so persistent. Once they enter the cell, obviously you even if you give drugs, they have to enter the cell. It's not like if they're not in the extracellular space. So within the cell, they use the cellular mechanism. In the cases of viruses, they use the cellular mechanisms to multiply. They enter the nucleus. They use the protein synthetic mechanisms of the cell and replicate in large numbers. So against these intracellular organisms, we do have one arm of the innate immunity which is quite active, and that is the natural killer cells that we talked about. These NK cells lie in the gray zone between innate and adaptive immunity. They do have some cell surface receptors, but are not actually active like T cells. They are cytotoxic T cells, which are not target specific. They can, once activated, they can lyse any target cell. So when there is ingestion by macrophages or entry into the macrophages by these intracellular pathogens, there are releases of IFN gamma and other cytokines, and these activate NK cells, and the NK cells then in turn kill the infected cell. And this is a very important kind of immune activity against viruses. So NK cell activity is very high in the early phases of viral infections. But soon enough, they cannot handle all the infection, and therefore the adaptive immunity has to kick in. And here the adaptive immunity, both the CD4 and the CD8 populations have very important roles, because the CD4 T cells, the macrophages, or which are infected by these organisms, they will then send out some of the proteins that are synthesized on their cell surface, which is then recognized by the CD4 cells and the CD8 cells. So the CD4 cells are inflammatory, pro-inflammatory. They secrete a whole spew of cytokines, whereas the CD8 T cells are cytotoxic and they lyse the infected cell. So effectively between these two, the infection should be taken care of. So if you look at what happens initially, the antigen presenting cell activates the T cell. If it activates the Th2 cell, it will help in the production of B cells, but this pathway is not very active. This production of conversion of B cells into plasma cells and antibody production has very limited activity, because the bacteria or the infection is all intracellular. But the presentation of the bacterial or the viral peptides on T cells then activates the cytokines. Now, these cytokines may be efficient in actually killing the infected cell itself. And if it does that and is able to also get rid of the infection, then obviously the host is the winner. But when these cells are lysed by the cytotoxic T cells, they may release the organisms which then go and ingest and enter a fresh set of cells, that thereby perpetuating the infection. Now, the classic example of a intracellular pathogen is, of course, and the one that we are most familiar with is Mycobacterium tuberculosis. Now, mycobacteria are phagocytosed by the macrophages and the dendritic cells. They multiply very efficiently within these cells, and if unrestricted multiplication takes place, then the cells themselves get lysed and release larger and larger numbers of the infective agents, the mycobacteria, into the environment. However, they also present cells to the CD4, antigen to the CD4 positive cells, which induces an IFN gamma response, and these activate the macrophages to produce antibacterial enzymes, which then kill the mycobacteria. So actually it is a tussle, literally a battle, as to who wins. Does the macrophage activation surmount the infection, or does the infection surmount the macrophage and kill the host and perpetuate infection? And in doing this, the CD4 pathway, you can see here, there is a complex set of mechanisms. Initially, the IL-12 is released, which then binds to the CD4 IL-12 receptor and stimulates the production of interleukin-2, which has an auto-stimulatory activity and stimulates more and more CD4 T cells. And there is production of IFN gamma, which then goes and binds the IFN gamma receptor on macrophages and activates macrophages with the release of various cytokines. So that in this mechanism, it's all a question of how successful this activation of macrophages is in dealing with the infective agent. Now, at the mucosal surface as well, there is a large number, I mean, as we are very familiar, a large number of infective agents enter through the lungs or through the gut, the vaginal tract. HIV, in fact, enters through almost all the epithelial surfaces. And then there is a mechanism which is more active through the interleukin-23-17 axis. So the dendritic cells of the mucosa are activated and they stimulate the production of IL-23, which then activates resident memory Th17 cells, and these then transform from naive Th17 to active Th17 cells. They produce IL-17, more and more IL-17 and IL-22, and stimulate the epithelial surface to produce antimicrobial peptides. So these then peptides can neutralize and try and kill the infection which is at the mucosal surface. Okay. They also produce various granulopoietic and chemotactic factors which attract, they do a number of things. They go into circulation, they reach the bone marrow, they stimulate the bone marrow to produce large number of granulocytes. There are chemotactic factors which then bring these neutrophils and other cells, granulocytes, to the site of the inflammation, and they then again phagocytose these organisms and destroy them. So immune responses at the mucosal surface, even for intracellular bacteria, is slightly different than at in other sites. But in an intracellular pathogen, the balance, as you saw here, there is a tussle between the activated macrophages killing the organism versus the organism overcoming the host cell and spreading and multiplying and establishing itself. And this is a very fine balance. Now, when this balance, neither side wins, and they strike a kind of a let's live with each other situation, that is when we have the dormant tuberculosis granulomas, which are lying somewhere encapsulated. The early phases, there is an immune response. It is maintained by low levels of release of tumor necrosis factor and IFN gamma. There is small amounts of chemokines which are released. Some monocytes, macrophages keep entering. There is a continuous struggle between the bacteria and the macrophages and the neutrophils. Some get killed, some overcome, and they live in kind of a harmony. This harmony is broken when there is another illness, a disbalance, any kind of therapy which destabilizes the immune system. There is, for some reason, there is a lowering of the immune system itself. Then the balance is tipped and the dormant tubercle bacillus then could activate and become a full-fledged tuberculosis infection. Now, the other is the immune responses to the large number of viruses. Right? So viruses, again, there is antiviral immunity, and there is, as we said, NK cells. NK cells are cytotoxic cells, and therefore they have an important role to kill infected target cells. The production of the innate immune response, the early response, which leads to the production of type 1 interferon. This type 1 interferon is a very potent antiviral agent. So the innate immune response has a role in most viral infections. However, how successful it is maybe milder cold, self-limiting viral infections. Most of the time, the virus establishes itself within the cell, and then you need to have a robust adaptive immune response. So viruses enter cells through cell surface receptors. They utilize the plethora of receptors on the cells, and then the antibodies that are produced as a response to the infection, they coat the virus and neutralize it and do not allow them to enter the infective agent through the specific target of entry, right? So these are what we call neutralizing antibodies. These antibodies are targeted towards those viral structures which are binding to the entry receptors on cells. So this is the only way of neutralizing the virus before it enters the cell. And this is the most potent way of dealing with a viral infection, right? And we will talk about it later when we look at the role of vaccines, their role in vaccines. The viruses also stimulate a very potent, obviously they stimulate CD4 cells because CD4 cells help is required for a robust antibody production, and CD4 help is also required for CD8 activity. These cytotoxic T cells, they kill the infected target cells, and since if the host cell is no longer there, then obviously the virus does not have the machinery to replicate and establish itself. So this potent CD8 activity causes the gradual decline of viral infections. Now, the most studied or the most discussed virus till the last 6 months has been HIV, which has been with us since maybe the early 80s. So it's really not a very old infection. Something which in the earlier part of my life I never heard about. I mean, it didn't exist, but immunology, it's quite interesting because HIV came in and everybody knew that there's something called an immune response or a CD4 cell, because everybody had to get a CD4 cell count done. And for 30, 40 years down the line, now we know everything about the immune system because the coronavirus is teaching us so many things about the immunity. So in HIV infection, since viruses use the existing cell surface receptors on cells, different viruses infect different cells, right? So the respiratory epithelial cells, some are vaginal infections, some are brain infections because they are using certain receptors on neurons for entry, so they only enter neuronal cells. So it depends on the compatibility between the viral protein, the protein on the viral surface versus the receptor on the host cell surface. The problem with HIV is that the host cell is the CD4 positive cell. So what the virus does is uses the CD4 molecule to enter the molecule on the CD4 cells to enter the CCR5 receptor on CD4 cells to enter the T cell, CD4 positive T cell. And when it multiplies, it kills the CD4 positive cells. So as the virus establishes and keeps growing, more and more CD4 positive cells are killed, resulting in a lack of adequate number of CD4 positive cells, so that the host immune system itself is compromised. So here there is no question of an immune response fighting the virus, because the virus is actually eliminating that immune response itself. In addition to that, HIV also has, and this is also the case with some other viruses, it also has the capability for repeated mutations, and therefore every time it mutates, it changes itself a little bit. So all the immunity, even if at all it is raised against this virus, now it is another virus, then a new immune response has to be developed, then it again mutates and another immune response. So this cycle goes on, and the host kind of gets defeated because the CD4 cells are anyway low, and there is no robust immune capability of an immune response. And these various immune responses, repeated efforts are difficult to keep pace of, and there is a total lack of any kind of a response. So again, we will come back to this when we talk about a little bit about vaccines. So the part of the immune response which is very critical is what we discussed again the previous talk as well, is the unique features, which is memory and specificity. So each one of these types of immune response to various types of agents that we talked about ends up by leaving behind a specific memory for that particular agent, and this memory is very, very specific. Right? So if your immune response has been boosted with virus A, that will not work against virus B. So once a lymphocyte is recognized antigen, a foreign antigen, it expands to eliminate the infection and leaves behind this memory. And that is what you see when you give a first injection of any antigen. It takes a fairly substantial amount of time to mount a good IgG response, which could be anything from 14 to 21 days. But if you give the same antigen a second time, you get an immune response which is many, many times higher and which is very much faster, because these memory cells respond very rapidly, and they have tremendous specificity. So you can see here that if you give antigen A and primary and you don't give antigen B here, but if you give antigen B, A and B here, the response to antigen A is this much, but antigen B is like that of this. That is the primary response. So the secondary response is many, many times higher than the primary response. And this is the basis of our vaccination, right? So in the vaccine, what are we trying to do? A vaccine, what we are doing is to give that same antigen to a normal, healthy person, but in a non-pathogenic form, right? So that it is inactivated or in some way, or only the protein which is necessary for an effective neutralizing immune response is given, but the antigen is given and the immune response is allowed to develop. And at a subsequent exposure to the actual infection, since the body retains the memory, it will immediately have a secondary response and produce the protective immunity that is required for fighting that infection. And that is the basic fundamental. Now, as we all know, the most So the most successful vaccine, or the only one which has caused total eradication, has been that smallpox. But there are various reasons why that was a successful vaccine. For one thing, smallpox, all of these are important when we discuss a few at the end, a little bit about the COVID. Smallpox infection was restricted to humans. There was no animal reservoirs. Smallpox does not cause a latent or persistent infections. Once infected, people die or they got or recovered. Yeah. So it was a yes or no phenomenon. Once you recovered, you had lifetime immunity. The symptoms of smallpox were detectable. That is, anybody who got it got sick. They had the pustules. You didn't need a great diagnostician, and you didn't need an MRI or a CT. They were isolated. Whatever was happened, people way back, the first. I mean, we have historical records of smallpox epidemics going back to 2,000 years or so, and the recorded epidemics. If those of you who read some things outside medicine, there's this wonderful book called Decameron about the so plague and smallpox has been around for a long time. So the reason that there is a high fidelity DNA polymerase and variola viruses are unable to undergo antigenic variation to escape existing immunity. So it was a very stable virus. It caused a disease. It was easily recognizable. Once you got it, those who survived had lifetime immunity. So when they heat inactivated this virus and gave it as a small scarification to people, they got, I mean, an immune response, and you were protected for life. So you had to only give it once. Even I, as a child, remember I got this response, this vaccination. And from the first description of smallpox vaccine more than 200 years ago, we've had a number of vaccines, and all of them are various types of inactivated or attenuated organisms, except in the very recent past. So diphtheria, whooping cough, tetanus, measles, more recently hepatitis, pneumococcus, etc., rotavirus. But the earlier vaccines, especially the childhood vaccines, these were diseases which also occurred mostly in children, and they were all either heat inactivated or they were toxoid vaccines, and they gave very good protection. Okay? Now, the fact of the matter is that if these vaccines did not give more than 95% protection to young children, the public would never have accepted these vaccines. I mean, I would not submit a newborn baby to 3 injections if I wasn't sure that it's going to get a pretty good protection. Iffy things did not work. So initially, vaccines were those that gave protection in more than 95% of cases. And a lot of the earlier licensed vaccines do give that, whether it be polio, whether it be MMR, or whatever it is. The vaccines for the other conditions, other diseases, has not been so wonderful as oh God, what have I done? So the vaccines for other, especially the intracellular, tuberculosis has been a challenge. BCG has not been a very efficient vaccine, although we still continue to use it. And see the complicated immunology of the infection itself, one can understand why the vaccine has not been so easy to develop. Because the immune response in the host is very, very complicated. There is no all or none. It's not that you eliminate all the bacteria. It has the capability to stay inside, isolated in internal organs. What happens when you give, we don't know, there is no one molecule which is stimulating the protective immune response. So there are lots of issues we have not been able to find. Now, the problem with HIV is also similar because the virus mutates too frequently. And because it mutates so frequently, we're not sure that if we produce, we know for sure that all the vaccines which have undergone very expensive and extensive trials, none of them have been really successful. They've all given protection, maybe 50, 60% of cases, but we do not think that, we did not accept that exposing millions of people to a vaccine which only gave protection in 2/3 cases or less was really ethical. And since behavioral change could control the infection to some extent, I think the pressure for the HIV vaccine has definitely waned in the last few years. In addition to that, one must also remember that we have been successful in producing effective anti-HIV drugs, which allow people to live for many years without any symptoms. So what are the issues which are important for a successful vaccine? So for a successful vaccine, we need, So for a successful vaccine, we need a sound understanding of the natural history of the disease, right? As I said, smallpox, it was straightforward. It was an infectious agent, it was highly infectious, but had just one kind of a symptom, obvious symptom, which could be diagnosed. It was all or none, it killed or you survived. So you need to know what the infective agent does. And we knew that from whooping cough, we knew that from measles, we knew the natural history for many of these diseases. We need to know the pathogen really well. We need to understand it. These early pathogens, which were isolated long before molecular biology was a force, but the tissue culture was advanced enough to be able to produce these viruses in the laboratory in large numbers so that inactivated vaccines could be made. Then we need to understand the immune response. What is a stimulating agent? What is a neutralizing antibody produced to? Is there a T cell response? How long does the immunity last? Is the antibody neutralizing in nature? To produce this, you need to identify the relevant antigens and peptides, which are crucial for protective immunity. And this, whatever that it is, we should be able to produce it in large amounts. Then to test this efficacy, there should be some kind of an animal model so that we know that the vaccine is able to neutralize the infection. Finally, you have to establish in human safety, which we do in phase 1 and 2 trials, and followed by efficacy in phase 3 trials. And that is not the end of the story. Even after licensing, you need to continuously monitor, because you do not know how efficacious this is, whether the immunity will last for 1 year or 2 years or how many years. And at any point when you give it into large populations, there may be adverse events. Now, in relationship to the COVID vaccine, we have a number of problems because we do not know the natural history of the disease. We have no idea whether the antibodies, when they are produced, how long they stay, what is the peak response. We really have no idea about the natural history of the disease. And every day, those who follow the literature will realize that since February and March, when we were just packing our hospitals with ventilators, look at the clinical spectrum of the disease. Okay, it seems to infect every organ in the body. It causes the coagulation problems, it causes central nervous problems, and in relation to what Dr. Misra was talking about, it causes Kawasaki-like disease in young children. So is it kind of an autoimmune disease? We do not know yet. So we have the pathogen itself. Well, their modern advances in molecular biology allowed laboratories to identify, isolate, and sequence the pathogen within weeks, within days. So that is something which we have been pretty good at. We know very little about the immune response. Since the last 6 months, the literature is growing, the knowledge is growing, but I don't know whether wisdom is really growing, because every other day you have papers which contribute something which contradict something which came out last week. So we are still in a struggling phase. We have, because of the competence of of identification and our knowledge of previous coronaviruses, we have been able, using various kinds of computer modeling, etc., we have been able to identify the receptor to which it adheres and to the knob protein on the virus to which we need to produce the immunity or the neutralizing antibody. So that is a good point. Animal models are far and few between. Obvious to actually look whether the neutralizing antibodies are formed, you need to do experiments in monkeys. And finally, we have the establishment of human safety. A large number of vaccines are in the phase 1 and phase 2 trials, and a few have completed and gone on to phase 3. Phase 3, as you've already heard, a couple of days ago, one adverse event in one patient, and a trial is being held, has been put on hold. The Moderna vaccine, which is one of the advanced vaccines, plans to recruit 30,000 people. Now, 30,000 people, and they have, as of now, not even, I think, recruited 10,000. People have hesitation in volunteering for various reasons. So it's not easy to do a phase 3 trial. Many vaccines, by the time they're actually produced, have a problem that the disease itself has started declining, which is what happened to both MERS and the original SARS, the 2003 or 2002 SARS. By the time they came out with vaccine candidates, the disease had been contained by public health measures. And as I said, in HIV vaccine, the impetus for the vaccine is kind of declining because of the amount of investment, which has resulted in not such great success, and other forms of control of the disease itself. But here, already we have similar situation that in China, by the time they got their vaccines, they didn't have cases. So they are doing their trials in Brazil and somewhere else, Peru or Chile or I don't know, some couple of other places, because they didn't have enough cases. So there are many issues before a vaccine can really be. Now, also the question of licensing. As I said, childhood vaccines and vaccines as we knew them now, I was involved in some vaccine development work for hepatitis E at some point. You had to show 90-95% efficacy. But now we are talking about, FDA has already said that they will approve a vaccine with 50% efficacy, right? Now, we do not know what is the protective antibody or how much neutralizing antibody is formed, how long it will last, how long this protection will be, etc., etc. So I would say be a vaccine optimist, but be also a vaccine skeptic. If it doesn't come tomorrow, one should not be disappointed. We are scientists. We have to wait for all of the safety trials to be conducted absolutely scientifically and have the data. And in this respect, my final word would be that in spite of all the political pressure, it has been very, very rewarding to see that the major vaccine producers have come together to release a statement saying they will not even submit their protocol for approval unless they are absolutely sure that their phase 3 trial data on safety and efficacy is sound. So, I mean, we have to go by the science. I think that I'll close with that. And if anybody has any questions, I don't know how I'm going to take the questions, but
Thank you, madam.
thank you.
Now you should all realize that the wait was the worth, because madam is such a lovely teacher, and you can see how she made the difficult things so simple and clinically relevant to everybody.